Hello, my name is Kimberlyn Petty, and I'm going to be talking about ELISpot assays today. And they're pretty cool, and so I'm pretty excited to tell you about them. What is ELISpot? ELISpot stands for Enzyme-Linked Immunosorbent Spot, or ELISpot, and it's used to measure secretory activity of cells. And this can be used to monitor B and T cell immunity. A pretty common application of this is an NHP or non-human primate. We can measure the interferon gamma secretion of cytokines, and this is most common in T cells. And then to use this, we can use this as a tool to indicate bacterial and viral infection within the immune system. Pros of this assay is that it's very sensitive and it doesn't require a lot of cells to run the assay. And some cons is that it can be pretty labor intensive, as it can take multiple days to analyze this assay. So ELISpot procedure. So PBMCs, or peripheral blood mononuclear cells, will be isolated from whole blood or tissues per method from a species. ELISpot can be performed on freshly isolated cells or cryopreserved cells. ELISpot reagents can be purchased in a kit. The plate will be pre-coated with a capture antibody. And then prior to the addition of cells, the wells of the plate must be coated with the media, and this will contain the serum of the species in which you're working with, and this will act as like a block to avoid non-specific binding to the wells. After the plate is blocked, cells are added to the wells of the plate in various concentrations along the different wells, and this is usually done per a plate map that is generated. Along with the varying concentrations, there will be negative controls on the plate as well. And these are used to compare the negative control, so the lack of stimuli, along with the samples that are stimulated with whatever we're using to stimulate them with. The cells are incubated with the stimuli, and this just elicits a response from the cells of interest. So, for example, T cells, we'll be looking, like in this kit, for interferon gamma. And so whatever stimuli we use will kind of generate this response. So that way we can measure interferon gamma. A detection antibody is then added, and then an enzyme conjugate, and then the substrate. And this just allows it to be able to be read on the instrument. And it's kind of similar to ELISAs, like we've talked about in class. So like a sandwich ELISA, because you're just kind of sandwiching your cells and your antibodies of interest on top of each other. After the process is done, the wells are then analyzed to determine the overall production per cell. This is what it would kind of look like when we're analyzing the wells. We see in the figure on the slide a control sample on the left versus a test sample on the right. The control shows no spots, and then the one that's labeled the anti-CD3, we see spots, which are indicative of the CD3 cells that are stimulated with the interferon gamma. And so all those spots that we see are cytokine production, and so we can count all those spots and we can see how many cells are producing that interferon gamma. First paper, they were looking at tetanus toxicity in samples. So they had a medium control, which we can kind of see on the top. And so there's not very many spots to be analyzed. There's a little bit there, but not a whole lot. And then we can compare it to the ones that are infected with the tetanus. And so we see those on the bottom. And then we looked at both interferon gamma production as well as IL-2. And these are both cytokines that are produced. And so we are able to count the spots in the interferon gamma and IL-2 that are positive for the stimulus and compare them to the control, so that way we can see how much is produced and then how many cells produce that cytokine. This next article was looking at COVID-19, asymptomatic or mild cases of COVID-19. And so we see 2019, kind of at the start of the pandemic, we see our negative and then our positive for each of these different tests. And then in 2020, negative and positive. We see a very mild infection, again with our negative and positive, and then our more severe infection, negative and positives. And then we also are looking at the exposed family members and the negative and positive, and then they were able to be compared amongst each other. Here's another article is looking at ELISpot in comparison to flow cytometry cytokine production. Looks like in specific T cells. So these ones were stimulated with CMV or looking for CMV infection, so we can see the spots and the amount of cells that are producing that cytokine. And then as well as the HIV positive viral infection, so we can see how many spots are there. And then these are for like no stimulation. So we were able to compare each of these to each other so that way we can see without the stimulus as well as with the stimulus and see how much is produced with the stimuli and how many cells are producing that cytokine. Here are my references. I really hope you guys enjoyed learning about this. I think ELISpot can be really cool, really fun. It is labor intensive, but it can be very useful in both clinical and research settings. And I hope you guys enjoyed. Thank you.